Today is a very important discussion because we have somebody who is quite unique in the sense that he describes himself as a problem solver. And he has been focused on COVID and the COVID related pandemic outcomes since the beginning. We've got somebody who has fought tooth and nail to find solutions to how we can resolve and protect health. And I have the privilege to discuss with him some of the science and some of the things that he is working on. I think it's very important for us to listen and to try and see if we can piece together what his vision is for finding out solutions for your health. We're coming up as well, one of our very important conferences in the next few days, next week, Saturday, the 9th of August. And I want you to look out for this because we're having a number of speakers focused on the neurological aftermath of COVID-19. A very important discussion, and it will fit right in with this discussion that we're having here today. So without further ado, I'd like to introduce Joachim. Joachim, can you just give a quick introduction to the audience?
Yes, good evening, everybody, and thank you, Philip, for having me on. My name is Joachim Gerlach, and I was a retired businessman. I sold my company over 10 years ago, and I was living very happily on the Canary Islands in a beachfront home. But I was already starting to work on different problems of chronic health disease, almost like an epidemic scale that we saw since the 90s. And I started to finance as a philanthropist different projects that were working on glyphosate, EMF radiation, and other environmentally caused illnesses. And so when COVID started on day one, that is January 2020, I think around the 18th, we dropped everything. And ever since that day, we've been working 7 days a week, practically around the clock, on finding solutions for this wicked little pathogen that is giving people so many problems.
Yeah, this is important. I think that there's something that I captured with you that I think is important to get across. You are passionate about this because you have worked almost every single day in the background, doing the research, working on products, working on testing, doing research in labs to find ways to get people better. What in the world is driving you?
It's very simple. I don't see too many working solutions out there. I like data. I like documented results. And if you look at the problem as it is unfolding right now, we haven't even solved, or we are now maybe getting closer to resolve let's say, the basic problems. Persistence of these S protein, of this very toxic protein from the virus that is our organisms are not able to degrade and get it out and clear it. The microthrombosis and other factors that are the underlying factors. And in my opinion, it's necessary to remove these underlying factors first before you even attempt any therapy, because you can't paint on a rusty pipe without removing the rust first. Yeah,
that's a very good point, Joachim. Now, I want you to think about something here. You have been working through the medical science, looking and researching and focusing on it. Why do you think the medical community has been so slow to find solutions?
There are various reasons. One maybe could be time. Most people have to work to make a living, and they have a practice, and they're treating patients, or they work in the institution, university, and so they are not at liberty to do what I'm doing, and many of my colleagues also are not alone. And so that is one thing, because the amount of information that you need to digest in order to see the patterns is enormous. And so that will take, you have to take your time every day. It's 8, 10, 12 hours every day, just researching and thinking and talking to colleagues and discussing things and publishing and working in the lab and so on. So it's difficult. And then there's the compartmentalized healthcare system that is also being reflected in the research. Yeah? So it's a multisystemic disorder, and so it's very difficult if you are in one field to understand the overlapping synergistic mechanisms that are obviously here at play. So that is another big problem, why there is not so much success in this. And yeah, there are more, but let's better look at what can be done.
Yeah, I think it's important to consider some of those points, because what you're doing is quite unique, and I think you need to be given credit for the fact that you've been able to do this kind of research and focus in this way for such a consistent period of time. And here is the reality, a lot of people don't understand this, but even if the medical community suddenly decided, you know what, today we're going to give it all of our attention, they still cannot recover the experience, the knowledge, and the observations that occurred earlier in the pandemic. It's not so easy to catch up.
Yeah, I mean, the database that is supporting our research is way more than 12,000 papers that are relevant to what we are doing. On the side, on one hand, understanding the disease mechanisms, and on the other hand, to provide data, look at data, document everything that can be done to mitigate the problems. Yeah? So it's a huge amount of knowledge that has been assembled. And now with AI, it's getting simpler, but the problem with AI is you can't really, not yet 100% trust it. So you might do a good job, 95% or 90%, but the 10% that you get mixed into this will make the whole result invalid. So it's still too early. Maybe in a year or two, AI will do a much better job than what I'm doing or other people are doing. But till now, it is the hard work, reconnaissance by foot, walking.
Yeah, absolutely. So one of the big issues, and this is the controversial bit that we have to touch on at the start, you're focused on the spike protein and the fact that the spike protein seems to be driving manifestation of disease. One of the issues that you are going to have is that many people are struggling to counter what they perceive as vaccine misinformation focused on the spike protein from the vaccine. And what I've observed is that the scientific community is happy to acknowledge viral infection as an issue, but not necessarily to focus specifically on the spike protein, even from the virus, because it has a potential correlation to the spike protein from the vaccine. How in the world are you planning to get around that block?
First of all, I have two principles: never assume, and second, don't fall for any narrative. Science is not a democracy. So scientific work is about evidence and about repeatability of results. And so I don't care what the narrative is. If the results and the science tells us this is a problem and this not, then I will have to probably follow the science. And in that case, it's difficult because there was a lot of science I call jukebox science. That means there were industry interests that were driving institutions and universities and the press to, I wouldn't say fabricate papers, but I can also I can manipulate the outcome of papers. I could even do that myself, which I will never do. But I mean, that means that the information available is not always trustworthy. And so I think we should all take a step back from narratives. Also, the people that are what we call anti-vaxxers, I find that they have been over the past 3 years very focused on just the vaccine injuries and the spike from the vaccine. And our research and my research and the lab tests that we did show that there is also a lot of spike from the virus, and the spike from the virus can also be extremely dangerous and toxic. It's the most toxic part of that virus. And there's other toxic ingredients also in the virus itself, but that's a different topic. So I would say that I'm not on either side. I'm actually sitting between the chairs, which is sometimes very problematic. But I think that the science speaks for itself, and I can only provide results for the patients when I stick to the science. Otherwise, I will be narrative driven and I will run in the wrong direction. Like many colleagues I watch and I go like, what are you doing? Yeah? Don't follow narratives, follow the science.
Yeah, this is a very important point. Now, when we think about the spike protein, and just to be clear with everybody, I am completely on board with regards to the spike protein. I've been focused on it since 2020. I think is a driver for immune and autoimmune responses. So the spike protein, I think, is an exceptionally unique and dangerous piece of this virus. And so I agree with you, Joachim, that we have to find ways of helping the body to address this. So I'm going to bring up your slides here and just take a look at some of the points that you're making with regards to the spike proteins. And this is quite a lot of information, but we're going to dissect it because it's so very important. Let's look first. You've got all of these papers. What is this about?
That is just for people that want to look at our work. This is my ResearchGate page so that you can see what we have published. We also did a lot of our own lab research, be it preclinical or clinical and in vitro, and a lot of in silico studies. I have a lot of work that I didn't have a chance to publish. It's just like we're under such time pressure that sometimes I really can't make it to publications. So these are 15 publications, but they give you a good overview. They go over the whole range of problems from cardiovascular to thrombosis to neurological disorders and other topics, antiviral, inflammation, cancer. And so it's covering already a nice range of topics that we are confronted with.
Yeah, so I think that when we think about this, because you've done a lot of research in labs as well.
That research, that particular research is of course more focused on, you know, people talk about supplements, and supplements are different than chemical drugs because these are natural molecules and our organism needs to absorb them, metabolize them in the gut, involving even the gut bacteria and different membrane penetration enhancers and so on. So my focus also is to improve and constantly improve the bioavailability and the efficacy of natural dietary supplements. That is what you see here.
Well, a lot of people are having a lot of negative connotations with regards to supplements. There's a lot of challenge that they're not as well tested as pharmaceutical drug therapeutics. What is your view with regards to how valuable some of these supplements can be compared to somebody having a tablet from one of the big drug companies?
Yeah, I had that discussion right at the beginning when I met Shankara, and we were looking at all the different mechanisms and pathways that are off. And then I said, okay, let's talk about it. So we started with this, with let's say the mast cells, and he said, yeah, let's take Montelukast, an H1 blocker, and then we take dexamethasone for the inflammation, we take for this, and then when we come from the 350 pathways, when we were at the number 10 or something, Shankara said to himself, oh, that's going to be difficult because we're going to have more problems with the side effects of the medications than actually we will resolve by taking those. And the problem is that pharmaceuticals tend to be very narrow. And we're looking at a multi-systemic disease, and you'll see later on why we chose the route to take dietary supplements into the equation. It doesn't mean that it's a question, this either or. I think it's a question when and for what purpose. So, of course, there are excellent drugs out there, and they work good, and they work fast, and they work very strongly, but they are very targeted. And then if you're looking at so many, I mean, we have some papers here showing 15,000 epigenetic dysregulations after exposure, 15,000. Now, so you have to do both. Now, of course, we see Beate Jäger and other doctors that we collaborate with, they use both. They sometimes have to stabilize the patient, even Shankara, reset the immune system in a way, etc., etc. So I'm not even engaging in that discussion, what is better. I would say you have certain uses where a pharmaceutical is excellent and a good choice, and other use cases where you want to have dietary supplements with a broad range of activity that they can help the organism to get into a self-regulation.
One of the challenges, and this is what you'll come across with the medical profession, and part of the reason why they would add some resistance is that they would say anything that changes the physiology of the body could be considered to be a drug or a therapeutic. How do they know how well certain supplements interact if people are already on drugs? And this is one of the things that, from a medical point of view, will make them quite hesitant.
I agree. Yeah, there are certain enzymes that, for example, degrade certain pharmaceuticals, but also dietary supplements, CYP450, for example. So if that is suppressed, that means then the drug the degradation of the drug will be slower, and it will stay longer in the blood or in circulation. And so if you keep up the same dosage, you might overdose. So if you have critical medication like beta blocker or other medication, then you want to be really careful by mixing it. Maybe you can also, like we do now with the Shankara, we have an alternating regime. Now you have a pre-treatment with pharmaceuticals that will stabilize the patient and enable the organism that it can use these. Look, a dietary supplement is not a treatment. It's actually providing the organism with what it needs to deal with the problems by itself in a natural, healthy way. Now, most people don't understand that. It's a different approach.
One of the things that I remember in one of the conferences that we had, I can't remember the name of the professor, and he said something that stood out to me, where he said that the way how the body deals with natural products is very different to how it handles medication, because it's treating the medication like a toxin, and so it will be trying to deactivate it as quickly as it can, as opposed to some natural products. It will treat it in a different way, that it maintains relatively consistent levels, making it far more difficult to low or high dose interactions. Is that a good point?
Yes, that's one of the arguments, but actually there's a better one that I usually throw at experts for pharmacokinetics and pharmacologists, and I say, look, you have here a cell, can be in different conditions. It can be ill, old, shortened telomeres, can even be a cancer cell, and we have the same cell type from the same tissue that is in good condition, and these natural molecules meet this cell, and depending on the condition of the cell, they can either kill the cell and bring it into apoptosis, or make it stronger and healthier again when it's worth saving it. So this you don't see with chemical compounds. And so when we get to that later in my presentation, you will be really surprised what happens when the organism ingests these kind of molecules. I mean, they're every day in our foods. So our bodies have been familiar with these compounds since day one of life. So it is a natural thing. You just have to write composition so that the organism can use them to get into a healthy state again. And that includes then, of course, things that would be deemed pharmaceutical effects, but it's not a pharmaceutical effect. It's actually the organism healing.
So it leads us into this picture here about blood levels in animal trials. What is this really about?
Now, look, before you start even doing any preclinical or in vitro trials or whatever, you should have your stuff together. That means you have to ensure that the molecular size, the dosages, and the way that we are working with liquid products, there's a reason for that. Why don't we go the cheap way and use just like capsules like everybody else? Now, it makes it very expensive and difficult to manufacture, difficult to transport. So we are not stupid. So why are we doing that? Because we want to ensure that these molecules are as bioavailable as possible. And in order to do that, you have to do a lot of measurements. And so you have to work your way through that. So we did actually, I don't know how many of these different trials in animals, having them ingested, look at the blood, look at cell cultures. If you look at the next slide, you will see that we have then induced a myocardial infarction in Widstar rats with a isoproteinorol, it's a catecholamine. And you can see here that we were looking, that was not actually a trial to see if we can tackle myocardial infarction. It was a trial to see how well can these molecules now arrive in the, for example, in the myocardial tissue. And you see the upper row was untreated animals, then the second one in the middle was like just conventional powder supplements, same dosage. And then you see the lower row, you see what happens when you do what we did. And now we're in the fourth generation, so even getting better with less dosage, better results. But you see the markers, the inflammatory markers and other markers for organ damage were up to 20, 30, 40%, almost, not 30%, more effective in the reduction of these inflammatory markers and organ damage markers. And
it raises an important point, because you've been focused, and maybe because you've had the capability in terms of financially, as well as the background in engineering and the detail, to be able to test a lot of these products. That's different from most things that we see commonly out there, isn't it?
Yeah, as I was not familiar with that industry, I didn't care. You know, we started to work especially in India, with experts for Ayurvedic medicine, for natural products. And actually you have even a ministry in India for natural treatment. I find that very intriguing. And so there's plenty of real good scientists, I mean, excellent scientists that you can work with in laboratories to see, is that a valid approach you're taking with the route that we did here. And it seems like, and so with an engineering background, you always try to then optimize and make it better. Now, so you ask questions, say, okay, what happened when we reduce molecular site? What can we micronize it? Can we put into lipid nanoparticles? Can we make it liposomal? Can we do it liquid? What kind of membrane penetration enhancers do we use? And now we're in the fourth generation. Now we can get these results with a quarter of the dosage.
Yes. So in this case, this was looking at the damage to the heart and the protection that came from one of the specific products to demonstrate its benefit over other products.
It's the same mixture, it's the same ingredients, even the same raw materials. That you see left is untreated. So you see what you see also in many vaccinated people, the infiltration of lymphocytes into the myocardial tissue, causing a lot of damage and fibrosis in the heart, and maybe even heart attacks, and if it gets out of hand. And in the middle of the slide shows you conventional powder supplements. They are helping. I don't say that they don't work, but I was not happy. And so we kept working until we had the results, what you see on the right slide. And then we said, okay, now it's a go. Now we can go into all the other testing and toxicity and other preclinical trials, and then ultimately also into clinical trials in the end of 2020 and the beginning of 21.
And this is a global collaboration, isn't it? Meaning that you're working with people from all over the world. And I know I'm in there as well, because we have been looking at the science since 2021 probably, and so it's quite a global sense and feel as the scientists meet together. I think most of these people I've had on in some of the conferences, looking at different approaches and so on. It is quite a valuable thing, isn't it, when you have this many people that you're interacting with? How valuable have you found it in the research?
Yeah, it's with everybody on that slide you see there, to some degree, are working almost either on a weekly or daily basis, or even in direct collaborations, in research collaborations, in publications, in lab research. And so, each one there you can see has significantly contributed either to treatment or to development or understanding the disease and other discoveries. And so it's a great honor for me to be part of that and to be able to mediate between all these groups. I'm kind of like the coordinating a little bit between all these domain experts. So that is a very rewarding work. It's a little bit tiresome, yeah.
Sometimes.
because of the
I'm
time.
just looking through all these conferences that we had done. These are incredible conferences,
Yeah,
huge amount of information from all these experts. I think one of the biggest ones was in 2022,
yeah.
where we were focused on multiple speakers, presenters. So this has been a journey of science, and people need to understand it's not just randomly one person working. It's a collaborative effort to try and find solutions that will hopefully move us on to a better place. But you believe that we have to tackle long COVID and post-vax issues. Is that what does this image mean?
This is a message to all the doctors and therapists, because when I talk to them, they say, "Oh, I have a patient in front of me, and he's developing diabetes type 2, and he has a brain fog, and he can't climb the stairs anymore," and they still have not understood and made the connection to either the injection or the infection. Yeah? And so we need to give all these doctors and therapists tools at hand so that they can understand and diagnose and find out what's wrong with that patient. Is it related to the virus or the injection, or is it just a normal, conventional chronic illness to start with?
So when we think about that spike protein, where is it? What is it that you want to say to people about this protein?
That it is a very tricky little bastard. The whole virus and the spike protein, it doesn't matter, even fragments of the virus, they are actually really good at switching off any host defense against jumping and infecting new cells, or in trying to degrade it. So the spike doesn't want to go. It is a very persistent, smart bugger, and it switches off anything that can become dangerous to its existence in our organism. And that's why it is persisting for a very long time. And especially the spike protein, there is more than you can read on what kind of problems it can cause and where. So what we do today, where it can persist, and in which form, in which cells, and then what to do about it.
One of the important things that I think is critical that stood out to me in the research is the fact that the spike protein can bind to CD147. And this is on immune cells. So when we talk about persist, gets inside immune cells, in effect, if an immune cell lives a lifetime, it will almost never get rid of it, potentially. That's how significant it could be in terms of sticking around in the body. So what does this mean?
Now, yeah, the first thing, the discussion we have, again, sometimes narrative driven, is now everybody's jumping on the S protein from the injection. And rightfully so. Yeah, I agree that this injection was a very bad idea, and that it causes actually sometimes much more aggressive and worse disease expressions than you usually see from the infection. But okay, that aside, though, the question is, can it be found? So now you see a lot of labs popping up in Germany and other places, and they claim that they can find spike protein. And the only person that I know myself very good and I trust to distinguish between the spike from the virus and the spike from the vaccine is Carlo Brogna, because he has done that a long time ago. And what he did is he was looking for a certain insert in the vaccine spike protein, which is a double proline insert, which is supposedly stabilizing the spike protein, and that is unique to the vaccine spike. And he found that after more or less like up to 6 months, he would find in 50% of the samples analyzed, he would still find circulating spike protein from the virus. So that is of great concern, because we get to that later. If you find spike circulating in the blood, it is a very sticky protein. It will not circulate for long. That's another misconception that people have. The spike protein is has a very high affinity for our host cell receptors and heparan sulfate and other substances that are especially within our endothelium. And so it will not circulate for long. So if you find free spike like Carlo did, that is suggesting that the transfected cells continue to produce spike.
No, I wanted to target something here because there is a one of the points here that is in one of the slides is that there is a perception that the only people who have symptoms or the people who have circulating spike protein are people with long COVID. What is the reality about people who appear to be asymptomatic and still have circulating spike protein?
We'll get to that later. I have some slides showing that. But the let's say why I showed this example here is now that is a second case report where you see spike from the virus, and you have a lot of papers right now, even from very distinguished scientists and institutions, and they say, we found spike protein after 2 years or 700 days in patients, and because we didn't find nucleocapsid, it has to be from the vaccine. And again, the road to failure is paved with assumptions. Don't assume. And in this case here, it shows that they could find very high levels of spike protein up to 10 months or longer in long COVID patients, but almost no nucleocapsid protein in the blood. So I caution everybody, don't assume, be sure, do it as Carlo does, look for the double proline, and then you know that's from the vaccine. Most vaccines.
One of the interesting things I've found in the research is that people will get long COVID or chronic symptoms, depending on how you want to look at it, from mild infection, okay? Meaning that they didn't have a massive serum viremia where the virus is necessarily circulating all through the bloodstream. They sometimes only had a little bit of a sore throat, a little bit of a cough sometimes, but yet they end up with long-term symptoms. My belief is that the lining, the epipharyngeal region of the upper airway, if that becomes infected, can then become a source and a nidus of ongoing circulating spike protein. And in that case, you wouldn't see nucleocapsid because it didn't really get in the bloodstream. The spike protein is though located somewhere directly in the body and is then being circulated exosomally. Any thoughts on that?
Now we have already the evidence. It depends also a little bit on the variant. So lately we see, we are working with Brigitte König, professor in Germany, one of the most distinguished molecular biologists we have here, an expert on infectious disease, mitochondria, and she set up a year and a half ago the first spike diagnostics laboratory, and she can distinguish between different hangouts where the spike is. So she has a lot of experience, and lately we have observed, because I saw that together with her, that we see a lot of exosomes circulating the spike, and it's not a vaccine spike, it's virus spike. And so the question was, how can that happen? And the best hypothesis so far is that it is a lingering, smoldering, in replicable virus somewhere in the organism, and the exosomes are shipped out with a spike because that's one of the most present antigens in the cell, in the cytoplasm. We get to that later. I have several slides on that. Yeah.
So I'd like to ask a question on that, because one of the challenges is that a lot of people who are struggling with long-term symptoms believe that they should be given antivirals to try and resolve it. And when I had looked at the research initially, I thought to myself, I thought that this was not a actively replicating virus, but viral remnants that are being replicated, which is different. Meaning, therefore, an antiviral like Paxlovid would not impact on that. Any thoughts about that?
It's more intricate than that. And antiviral theoretically will, of course, address some of these problems. But I'm not sure if it would get to the place where the virus is persisting in the first place. And second, it will not solve all the problems, because we better go through the whole presentation, and then you see why I'm saying that. So and with all due respect, look at the publications lately on, I don't want to say any names of pharmaceuticals, but this approach they did, the 3CL main protease inhibitors, first of all, is already, it's coming in very late. That means the virus is already in the cell replicating when you start to go for the main protease. And the main protease was our first drug target, yes, in 2020, in 27th of January 2020, already identified. But what they did now is that by doing that with a targeted pharmaceutical, they have succeeded to push that 3CL protease, which was conserved over decades in all coronaviruses. They have ramped up the selective pressure, and now it has mutated. So one of the most rewarding drug targets, congratulations, now they have gotten it to mutate. Cool. Okay, let's go on.
So when you looked at this paper here, looking at the spike persistence again, this is just really emphasizing the point that in some people, there is a nidus of ongoing spike protein. Why does this not necessarily happen in everybody?
Who knows? Look, when we finish today, you will understand why nobody has been yet successfully being able to determine how much or what is the percentage of people in the general population walking around with persistent spike protein. Nobody knows, not even we know, until we start now our big survey. By end of this year, I can tell you, hopefully.
Yeah, these are important points because it has such a relevance in terms of what goes on. One of the things that we had done a paper on and we'd published about the long COVID picture was this persistence in the gut. And from a clinical point of view, I'd observed that the people who seem to have gut issues were also the same people who seem to be affected by long COVID symptoms down the line. And then this kind of paper will fit into that kind of picture. What are you looking at here?
Now, this is another battleground of persistence. And our dear colleague, Dr. Carlo Brogna, has not only discovered the double proline spike insert in the circulating spike after vaccination, he has also discovered before that SARS-CoV-2 or the virus can infect and replicate in our own bacteria, behaves like a bacteriophage, so to speak. And that is making the problem more complex because now the virus is not only infecting our own host cells, it can also replicate in a different species, so to speak. We have 35 trillion bacteria and 35 trillion cells ourselves. So it's a balance of numbers. And so it's another reservoir, and that plays a role in transmission. It causes dysbiosis, causes gut inflammation, and a lot of other problems, but we can't really get into all that today. But if you want to get rid of spike protein, you have to take this mechanism into account, which I will not address today because the time will not allow. It can be done with antibiotics, has been published. Carlo just sent me his new release paper an hour ago, where they even did it in prenatal conditions successfully. So you can take amoxicillin and rifaximin, but if you go back to that slide, I show you something else that is really important. If you now look at the battlefield here, it's a Stalingrad condition. You have you have a two-front battlefield here. You have the epithelium in the gut, which is also full of spike and virus persisting there for a very long time in the epithelial cells. And you have spike and virus in the bacteria. And they are located very close to each other. So when you do an intervention targeting the bacteria, you should, it's recommendable that you also at the same time see that you can detox the epithelium, which is right next to it, so that you get it all out at the same time. Otherwise, as it is a virus and it's infectious, you can run into a so-called ping-pong infection between the bacteria and the epithelium if you don't do a good job here in clearing it all out at the same time. So we get to that later, how that can be done.
Yeah, these are very important points, and it demonstrates just how complex this process can be. I'm trying to think of a good example, and it's almost like chelating heavy metals. If somebody had lead toxicity, you have to be so careful as to how you remove it. It's starting to fit into that kind of picture. But let's move on. What does this study indicate? What were you looking at here?
We're going now through different body regions where spike protein and or viral persistence is of concern. This is a PET imaging of a head, and you see there on the left, you see what is interesting to me is the persistence of viral RNA and spike in the nasal region. Because it is not only causing inflammation and neuroinflammation and can not only travel to the brain from there, it also has a second big effect, and that is to the arsenal of SARS-CoV-2 spike belongs also the suppression of type 1 interferon. And so as long as it's present, this type 1 interferon is suppressed. And if you talk to any real good virologist, he will tell you our first line of defense and the most important weapon we have against any kind of pathogen or viral or bacterial infection is type 1 interferon, especially in our nose. And so the persistence of spike in that region will make us vulnerable to opportunistic infections, which we
I'm glad you mentioned that, because this is part of an important piece of work that I had found as well. And Omicron was found to be 7 to 10 times more preferentially infecting these sinus regions.
Yeah.
And what I think we're starting to see, I mean, this is just anecdotally, is a lot of people with inflammation in the sinuses without any symptoms. We see it on CT and MRI scans as a routine. I now know to look out for it because it is such an important piece of information. But this paper here, similarly, what were you looking at with this?
No, that is the result when it travels through the nasal cavities and the olfactory system into the brain. And there was a, as you can say, dead people don't lie. So these were pathologists examining randomly dead bodies in Germany, and they found in 60% of all the people that had spike. Give me a second. Yeah. I hope he's going to stop. Sorry about that. One moment.
Okay.
So the findings were in the examined corpses that did not die from COVID. They were randomly sampling cadavers, and they found in 60% of the ones that had COVID before, they found a lot of spike protein in the skull bone marrow and in the meninges, which is right next to the brain. And so that is of great concern, because you don't want to have such a toxic inflammatory protein so close to your brain and in that region and persisting. So that's why I was mentioning it here. And on the next slide, you also see some more detailed pictures of, and they talk of enormous amounts of spike, not just a little bit. And that is a very delicate region.
And these were not necessarily people who had COVID-related symptoms. These were just.
You can find a lot of people that have no symptoms at all, and when you do a brain scan, you can find that the blood circulation and the blood flow to the brain is severely impaired, and you can also find lesions in the gray and white matter in completely asymptomatic young people that have otherwise no other symptoms, or they had even an asymptomatic infection. So
yes,
the data speaks for itself. I mean, I don't want to create unnecessary panic towards with the virus, but this virus is a real bad pathogen, and it is not only the injection that is dangerous. Also, we see a lot of patients that never were having an injection, and they are very, very ill, and you can see on brain imaging, you can see the problems and in other regions of the organism as well. So I would caution a little bit not to take that lightly.
And what was it that you were focused on here?
Yeah, it is a phenomena we see that with the brain fog and the inflammation, it cannot be resolved easily. And I was wondering for many years why that is the case. And our brain has a system of flushing. It's called the glymphatic system. That means at night interstitial fluid is being, so to speak, pumped through your brain and cleans out any amyloid, inflammatory molecules, and whatever is on as garbage in the brain. And the astrocytes are involved in that, and they actually provide the valves where this liquid can be let in and out from the brain parenchyma. And if you look on the next slide, you see why I'm highlighting this, because that's another place where spike protein likes to hang out, and that is the valves, the aquaporin 4 channels in these astrocytes that are opening up and closing and letting the cleansing process happen at night, for example. So these are all extremely delicate places where you don't want to have spike. Trust me.
Yeah, this is actually very, very useful, Joachim, because I'm just about to do a presentation on the brain fog mechanism and why I think it was happening. And part of the focus was not just on the aquaporin 4 pathways, but just to do with astrocytic inflammation as part of the whole picture with regards to it. This is huge stuff. And the problem is that when these things occur, they're not easy to resolve.
No, yeah, it depends. We get to that later. I'm just showing now why it's a good idea to get rid of it and giving some examples of places. For example, the parasites. Most people talk only about the endothelial cell. The parasites are actually, there's discussion about that, but they seem to be more susceptible to the spike protein than even the endothelium. And when the parasites get infected, you have another phenomena, because they contract. They become senescent and contract, and then they will cause vasoconstriction and permanent vasoconstriction.
Yeah.
And then you come back to the glymphatic system. Okay, there is no pump for that fluid to be pushed through the brain at night. Actually, the way it works is that our arteries are pulsating with each heartbeat. And when you sleep and you're in deep sleep, then this seems to be more pronounced in the brain, so that you pump the fluid through your brain. But when your parasites are full of spike, you are going into also arterial stiffness. That means you're not only closing the valves for the glymphatic system, you're also stopping the pump.
Yeah, you've just hit another nail on the head, Joachim. As it's one of the areas that I was looking at with regards to what I call the COVID storm, where people get reinfected in an immune primed environment. And when we saw the Japanese data on the tiny microscars, my thought was that this is parasite disease. Parasites are being damaged and then leading to microscarring. And so if it can occur in the heart, absolutely it can occur anywhere parasites are based, which is brain and kidney.
Yeah, I mean they're behind. They're actually in the layer behind all the arteries and the vascular system. But okay, let's go. We have a lot of slides to cover.
Yes, I know. Yes.
And that is, of course, here we have seen that slide probably from many places, like Professor Arne Burkhardt's findings. What you see here is a cut through an arteriole, a very fine, thin artery, and everything that's brown is actually spike protein antibodies reacting to spike protein being sitting in endothelial cells. And that is, of course, one of the main drivers and our main targets in the spike, let's say, helping the body to get rid of spike. Now, because that is lining all your blood vessels, and so that's one of the biggest organs you have. And if that's full of spike, it's going to cause a multitude of problems like inflammation, so thrombosis, and other really nasty things. And these cells reportedly turn senescent. Let me start highlighting that at this point here in the presentation. What is senescence? Senescence is a status of a cell, which I call like a zombie-like state. That means whenever a spike protein or SARS-CoV-2 virus, and also other viruses, belong into that category, touch on the membrane of the cell or fuse with the cell or intrude the cell is being switched into that state. Again, it's probably, we don't know exactly why that happens, but it's maybe protection from the body, and it can also be a safety mechanism for the virus to stop the organism from getting rid of it. Because it's like
so the cell is not dead, it's just not functional.
It is highly inflammatory. It cannot die. It cannot go into apoptosis, and it cannot. The autophagy is blocked, and it cannot replicate anymore. So that's why we call it a zombie cell. So it's easier for people to understand what we mean, and that is very widespread, much more widespread, which we will see right now with long COVID and post-vac conditions.
And so what does this mean? Does this tie in with it?
Yeah, we have to also keep up our sense of humor a little bit here. Otherwise, we're going to need a self-help group after we finish. So what happens when the spike protein in the virus touches cells? What happens? They fuse together. It's a so-called Syncytia formation. And that is like, you can have 20 or, I don't know, a high number of cells. They just fuse together similar to a fried egg. And so here you see you have still nucleus, the yellow from the egg, but the protein around it is it's toast. That's it. No chicken are going to hatch from these eggs anymore. They're dysfunctional. And that's exactly what happens when you are exposed to the spike protein and the virus, and also the injection, which is still up for discussion, by the way.
And so this is another image showing a similar pattern where you get these giant synthesias where everybody joins together as one large cell, but then becoming non-functional.
Absolutely. They are completely non-functional. They become senescent as well as zombie, and they're actually highly inflammatory, and they are an excellent no-go area for the immune system, and they are a hideout for the spike protein and the virus. It will sit there for an indefinite time frame, uncontested. And that's another example. You see here that it is clearly showing in many lab reports that this S protein can cause massive syncytia. Massive.
And in this case, worsening the heart failure.
Yeah, on the next slide, sorry, I mixed up some slides, doesn't matter. The same happens here. You remember Jasmine, our dear friend, patient from the UK that couldn't breathe anymore after exposure. She was part of that study where they were measuring the oxygen exchange in the lung, and it is probably caused also by fibrosis, but also by lingering syncytia. A lot of papers showing that senescence and these kind of conditions in the lung tissue itself. But most concerning is on the next slide, that is really, that woke me up a bit here, is that the SARS-CoV-2 spike can generate syncytia in the cardiomyocytes. Because what happens if you have one spike protein and you can fuse a large number of cells, that is like leveraging your damage potential. It's like a hand grenade. And it can be stopped by furin inhibitors. So I'm very happy that we were looking at that very early in our work, because it was supposedly part of the gain of function work that was done on that virus. Without a furin cleavage site, there would be no pandemic. So furin inhibitors, of course, do make sense, by the way.
And so in terms of that is very concerning, because one of the things that we can anticipate out of it is an exacerbation of heart failure conditions, where people's heart function will go down, not necessarily heart failure immediately, because these heart muscle cells are just not able to contract as effectively.
Yeah, and they're difficult to repair. So I would say, like, the crown jewels are our neurons and the myocardial tissue, and of course the progenitor and the stem cells and the ovaries. So these are the crown jewels, so to speak, that you really want to protect. And by the way, we get to the next point here. You can see even stem cells fusing together into Syncytia. That is.
And when you say stem cells, you mean the original grandfather producing cells that maintain the counts of normal cells in the tissue from the bone marrow. So they are getting destroyed.
They are getting destroyed. We get to that later. Just an example of a leverage damage, because with Syncytia you have one spike can kill or disable multitudes of cells in one go. That makes it more dangerous. You can skip the next 2 slides. Sorry, 3 slides. If you go there, go to that one with the pictures of the stem cells after stem cell transplant.
Is it this one here?
Yeah. That was also like an interesting paper here, where you see somebody had a stem cell treatment transplant, and he got COVID after that, and that was it. All these new stem cells were done. Look at that after the infection.
Now, would it be reasonable to say that if he had a stem cell transplant, he is likely to have a degree of immune suppression and therefore significant viral circulation, which would have exacerbated this? We wouldn't expect that in a normal person with a reasonable immune response.
Now, yeah, it depends, of course. If you look at the data, we get to that now. So the PBMCs and the immune cells themselves are having plenty of receptors enabling spike and SARS-CoV-2 to enter the cell. Then you have all the phagocytes. When they ingest spike or virus, they choke on it. Then you have some of the virus, as we saw in the spike, finds its way into the bone marrow, as shown in that other pathological examination of corpses. So if it goes to the bone marrow, then it's very close to the stem cells, isn't it? And there are a lot of research there. The resulting immunodeficiency is difficult to. That's another lecture for it by itself, because you have now from the bone marrow, you have the stem cells and the progenitor cells going through the cyme, being differentiated into different.
Mm-hmm. And what is this here?
Again, another example of senescent cells and long-term resistance to degradation by any kind of degradation mechanisms in the body to get rid of persisting spike and or virus. Very clearly here. Yeah. Mm-hmm. And they are senescent. And then you have here, okay, that's up for debate still, and I hope, okay, how can you say? I'm not really sure what to emotionally say. If senescence happens, of course, it's horrific on one hand. So if the messenger mRNA vaccine do cause pathological syncytia, then that is, of course, of great concern. But it also opens up a chance for us to nuke these cells with certain compounds so that they go into apoptosis and stop producing spike protein. So it's a problem on one hand, and it's also the Achilles heel of that little bugger on the other side.
Yeah, these are huge issues, and it can sound overwhelming to people. But one thing I would say about the human body is that it is remarkably resilient. Mm-hmm. And where we may think that there are weaknesses, it almost has multiple layers of failure. And even when some of these things happen, and I've seen it in real world where patients have, say, had a stroke, damage to a particular area of the brain, definitely scarred, but somehow that brain finds a way to function effectively even with that kind of damage. So we wouldn't want people to think that there is no hope. This is just about giving the body a chance to be able to overcome some of these challenges.
Well said, Philip. Yeah, because that is really the amazing thing. It all sounds, because we're looking now at these micro pathways and what's happening there. Look, even with EBV or other viruses, you have similar stuff happening, and people are still walking around more or less healthy to some degree. So we shouldn't overestimate these problems. They need to be addressed, and if this happens over a longer period, look, it's an intoxication in a way, as you mentioned before. So what are the hallmarks of an intoxication? It is the toxicity, the dosage, and the duration. Yeah. So maybe the toxicity is not as bad as mercury or uranium, but if the duration is long enough, no, then you can have problems with it. And so we have to see that we get this out better.
Absolutely.
And one way that is promoted is autophagy. If you go to the next slide, yeah. So autophagy is, of course, a legitimate route, but I'm not sure if it's going to work. I haven't seen data yet.
Well, can you explain it so that people understand what you mean?
Okay, in layman terms, like in my terms, that I would understand easy, is like you shut off the food supply to your city, you know? And then nobody gets any food for like 1 week. What would you do? You go into your basement, you go into your storage little room that you have, and you look at everything that you have to eat there, and you will eat it all up for sure. And that's what the cell does. If you starve the cell or the organism with fasting, it will start to look at all the stuff that it can scoop up and digest it, so to speak. Now that's what's happening here, more or less.
I see. No, that is a very good, that is a really good analogy. And so therefore, the cell, even if it has spike protein in it, because it needs to get energy and resources, will digest it just so, not quite?
No, that's the problem. If you go on the next slide, and that is the discussion we have, I mean, it doesn't hurt to do intermittent fasting. It is, of course, a good thing. But it is probably not enough to get rid of spike protein that's in these cells, and especially not if these cells are in syncytia formation and or heavily senescent. So I have my doubts that intermittent fasting will do the job. Maybe it does something. I don't know. We have to measure. Nobody's providing data. Everybody is putting out hypotheses without providing data. And so we are, I don't know if we can do also another arm. It costs money. You have to then monitor the patients and the diagnostics for each survey. If you want to do it well, the diagnostics alone are $6,000 or euro per patient. Yeah. So that is not something you can pay easily, especially if it's not a large number. So, and if you have to create another arm for another survey, then I would like to have people also put their money where their mouth is and contribute before we look at things. But
and so where are you targeting then? So if fasting can't do this, are you suggesting polyphenols?
yeah,
and what do you mean?
Hypothetically, it would be possible. Now, so this was a paper, a study done by colleagues that I don't know, and that are working on similar dietary supplements, and they are putting out the hypothesis that these polyphenols and flavonoids would be a very good measure to push autophagy. So if you can't get it done by fasting, there are certain pathways that you can push. So that's why I mention it here, so that we have to go now through all options. We want to get rid of these proteins. So autophagy, maybe put a question mark. Fasting, maybe with the help of from our friends here from these molecules, could be possible. Let's put it that way. Yeah?
So it comes into who then really is affected, because this is the other question: is a lot of people are fine. They're not concerned. They think that they're going about their living life as normal. Why should they be concerned if they have no symptoms?
Yeah, look, like I said before, you have a lot of diagnostic tools right now to show that symptoms can lie. I prefer data. Yeah, so if you look at brain imaging, for example, and you see that the blood flow is restricted, or if we went 2 weeks ago, we went to Dr. Jäger into her clinic, and she has this laboratory to detect microclots or microthrombi with the fluorescent microscopy. And so we were curious. It was quite expensive, and we just paid for it with 4 people, and we had our blood examined. And to our surprise, 2 in the group had extremely high levels of microclots. And that is what you hear from everywhere. So maybe you're asymptomatic, but if you're walking around with spike and microclots for a long time, then maybe get another reinfection, get another problem or any other health problem, or opportunistic co-infection with candida or whatever, and then you might have a problem. And that's what doctors are reporting. You see vaccinated, for example, being okay, having a little bit symptoms, and then they get reinfected, for example, and then all of a sudden they fall over the cliff. I mean, you're in clinical practice. I don't know if you see these kind of events happening. You know?
Oh yeah, yes. This is one of the big worries, is the presentation of disease, sometimes very close to infection, can be quite significant. People can get quite ill very, very quickly. And it suggests multiple layers of failure, because normally your body is quite resilient to quite significant infections, So if younger people, even if they got cholera, would survive it, sometimes even without intravenous fluids. But when you have multiple levels of damage already in place in the body, even the slightest infection can push people over the edge. And so this is, I think, a big contributor to illness and mortality across the population.
I mean, I did the numbers on the excess deaths in Germany just a week ago, because people were talking too much about, so many people die. It is significant. I mean, you can also look it up for every country. You look at the average number of deaths per year until 2019, and that was in Germany 930 to 940,000 per year. And then you look at the number of deaths 2020, 21, 22, 23, and 24. So for these years, it was in 2020, it was not as much. It was 50, 60,000 more than average. And in 21, it was already 100,000 more. In 22, it was at the highest, and now it's slowly again declining. But all in all, in Germany, the years from the day 1 of the pandemic until end of 2024 have cost 380,000 lives. With Germany with a population of a little over 80 million. So maybe percentage-wise, it's not so much, but it's, guess how many people died in World War II, civilians, through the bombardment of the Allied forces in Germany?
How many?
Exactly the same number.
Wow.
Yeah. So it is of concern, but it's getting better. So it's not that so much that people die like flies. It's more like people getting chronically ill and incapacitated.
I think the only thing I will say with that is that it's the principle of the mortality debt, meaning that the excess deaths are coming down, but when you look at it compared to the factors, to the amount of people who already had illnesses, were close to death, who had already died in COVID or related to COVID, the mere fact that excess deaths are not significantly below the trend between 2015 to 2019 is a very worrying trajectory, even though it is coming down.
Yeah. Okay. So, but let's do all within our power to help keep people in relative health and improve that. Yeah. So you have to go back one more slide, and then I can maybe help a little bit with the misconceptions that are around here. So if you look at the virus, it actually looks like a burdock, pretty much, and it actually works the same way. Yeah. So a burdock, if you remember how sticky those are, they don't fly around for long. They stick to fur, clothes, skin, hair, and actually the part of the burdock that is the most the stickiest part are these spikes of the burdock, and same like with the spike protein. So how can we expect these to circulate for a very long time in our bloodstream. And if you go to the next slide, you'll be surprised. There's 20 plus different receptors and co-receptors on the cell membranes of different target cells in different organs where the spike protein can fuse and bind and enter. So that is a lot of locks that this key is fitting in. Yeah? So that's a big problem. So of course it will not circulate for long. And that makes it even more difficult later than to find it. We get to that. And then you have, on the next slide, you have then a different process, and that is not a virus, that is a lipid nanoparticle that can go into any cell and convert that cell into a spike factory, so to speak. Yeah? And when doing that, the cell becomes dysfunctional, and a lot of spike proteins are being excreted into the bloodstream or into the interstitial fluid or into other regions of the body, and the spike protein itself then won't replicate anymore. Now, that's also important to understand. Spike doesn't replicate, it's not a virus. It will, but it can go into a cell and enter and stick around in that cell. So if you want to clean up that mess, on the next slide, you see what we are dealing with. Very simple. It's a tricky business. You have, on the left, you see productive cells, and on the right, you have depot, in German is deposit cells. That means the productive cells have been transfected by the lipid nanoparticle and are still pumping out spike proteins, or they are having a virus inside that is still active and that pumps out spike proteins as well. And the deposit cells mean there's just a spike went in there, they became senescent or dysfunctional, and then the spike sticks in there, and these cells become highly inflammatory. Yeah? So we have done a lot of research since years now on the different ways how the spike enters, how it sticks around, do lab research, and you can see that on the next slide. So we worked, first test was in India to see where we also did the bioavailability studies, and on the next slide you see we finally managed to combine spike protein with a fluorescent dye that you can see on a scanner. Yeah, so we wanted to first see what happened to the spike if you inject it into the tail vein of a mouse. And yeah, that is 72 hours. It looks like, I think I don't need to say any comment here. It is complete distribution through all organ systems. These were not humanized ACE2 mice, so they have no receptors. We just wanted to see first the distribution. This was the beginning when we started. And on the next slide, you see then we started to test some binders. That's how we started. We start first always in animals before we go to humans. We get criticized sometimes, but I don't want to start on humans. Let's start on mice and hamsters first. We didn't kill these animals. And so you see two animals here, both being injected, and after 72 hours, you see one animal, always on the right, gets treated. And this was just the first generation of binders. And we wanted to see, is it possible to bind the spike protein? I was not aiming at degrading it or dissolving it. That has different reasons. I prefer to neutralize it, bind it, and help the organism in a natural way to get it out of the organism. And you can see after 11 days, we saw already some significant results. The binders seem to work. So we got to work and continued. In the meantime, on the next slide, you see Professor Koenig had opened her laboratory in Germany. And so we had a run with patients that went there and looked at if they have spike protein in their blood. So as a doctor can take a blood sample, Vida's blood, in vials, send it to her lab, and she will then spin it and work on it, and she can detect free spike in the blood, spike in exosomes, or spike in immune cells. She can do more, but these are the standard diagnostics she's offering. And so we were very, very happy to meet her and be able to work with her now on a daily basis, so to speak. She became partner also in the development of new testing methodologies with me. And so now we are moving in that direction. And on the next slide, you see how that looks like. So if you take these blood samples, you see, for example, that was a patient report here on the 31st of October 24. spike protein in plasma, free spike was negative. And that is actually the majority. You don't find much free spike. What you do find is a lot in exosomes, as here. So 228 picogram per milliliter is quite high. Let's say the detection level is 4 picogram. Medium, we consider something between 50 and 70 picogram. And anything above 100 or 150 is already as high as it comes normally. And then you see here that was a patient that went into treatment here or took dietary supplements. You can see that then the exosomes went down, and then something strange happened. The immune cells, remember in October, they were negative, the lower one, the PBMCs were negative, and in November, they were slightly positive. Remember Bruce Patterson? He's going to kill me because, okay. These are the phagocytes. These are probably the monocytes and macrophages that were, we knocked out the cells with a spike, and they came and they chewed up the debris. And by doing that, they ingested the spike. Yeah. And that's
actually a very, very important point, which is that it's not good enough to remove the spike out of the cells, but you have to remove it and bind it immediately to prevent it from entering other cells because it is so sticky.
Exactly. Now we get to that. So let's look now at the different things that Bridget was documenting here. Look at the spike in the exosomes and extracellular vesicles. We mentioned already before. There's plenty of evidence here. You see, and it is mostly actually on the surface, on the membrane of the exosomes. Yeah, even many times showing the RBD of the spike being able to bind. But it's actually a little bit of concern because extracellular vesicles are kind of like lipid nanoparticles that our own body produces, and they can fuse with any cell membrane, same like the lipid nanoparticle from the injection. But be it, we have to live with it and get them out somehow, and it seems to work already. At least the preliminary data looks very promising. On the next slide, you find a surprise. Actually, the exosomes recovered from the plasma of COVID-19 patients, and the spike it was containing was a lot higher in mild patients than in severe patients. I don't know why that is. They don't know either, but that's what they measured. That's what I said. Let's look at data and not assume things and not be narrative driven. But the mild had much more spike circulating in exosomes than the severe cases. Very strange. I have no explanation for that.
That's interesting. That's something that is well worth thinking about. But yes, I'll reflect on that. That's an important point. I suspect it's the source of the exosomes.
Maybe somebody comment. Maybe somebody has an idea and can comment it on the video or on LinkedIn later.
Yes. Very good point. Yeah. So what is this here about?
This was a study done even by BioNTech, Pfizer. And they documented also that the spike is being shipped around by exosomes after transfection of the cell. Yeah. So it is something very clearly shown, and it's the majority of the spike we see in the patients. Majority is in exosomes at the moment.
And yes, I think it's such an important point that people understand that the cells communicate with each other. It's like sending a FedEx parcel to a relative. You package up something, put it in a box or a special envelope with bubble wrap, and then you ship it off so that another person can get what you have packaged. That's very much like the exosome. And what these cells are doing, potentially because they have too much of it, they are packaging up these spike proteins and saying to other cells, listen, we're struggling to deal with this. Can you do something about it? And that process is then transfecting multiple cells, because then if they get mRNA, they will make more, and the same thing happens and just keeps on going.
It can happen with mRNA. It is only documented to a limited time frame after vaccination. But yes, it can happen. They found them in exosomes, mRNA in breast milk and other tissues.
Yeah.
Mostly it is spike that is not, it can't reproduce. So the shedding is another thing that we have not really resolved yet. I'm not sure if it is spike protein because it's not enough. If you see the number of spike proteins we are confronted with during transfection or infections in the billions or even tens of billions, and how much could you shed by air through exosomes? Maybe it's in the tens of thousands, but it will not be relevant really. I'm not sure. It could be something else. I think shedding is existing, yeah, but I don't think it's the spike. But that's a topic for another day.
Yes, yes, yes. So tell us some more about this persistent spike protein in the white blood cell PMBC.
Yes, this is, I will show later a whole list of what is considered PBMCs. Yeah.
Yeah.
These are immune cells, roughly speaking. And so what you see, one type of PBMCs are the so-called phagocytes. These are actually the garbage eaters in our organism. So they can, and here's a fantastic animation of a macrophage trying to ingest SARS-CoV-2 viruses. Yeah? And I found that really interesting. And if you look at the next slide, you see, and that is what also Patterson has, we have to give him credit for that, discovered and documented and built a whole regime on it, diagnostic panels and treatment, is that the monocytes and the macrophages, when they ingest spike protein or virus, they turn also into zombies. They turn senescent. That's what Patterson has not yet understood, but he's hopefully getting there. And so that means that they are dysfunctional, highly inflammatory, and that's what Patterson described. He said they're like garbage trucks driving around the block and stinking and not getting rid of the garbage. No? And so I call them, because these cells are like Pac-Man. On the next slide, you see that they are eating pathogens and cell debris and all the garbage. I call them zombie Pac-Mans. Yeah? And they have to be removed. And that's what we find a lot in the lab, is these PBMCs full of spike. And to make it a little bit more interesting, on the next slide, you see that even these lymphocytes and PBMCs can be turned into Syncytia. So, and that's could be part of the microthrombi and the debris that you look at under the microscope, because when they build these cell conglomerates, they start to be of considerable size, so they can contribute also to the microclots, so to speak. You know? But that is of course, killing a lot of lymphocytes and the immune deficiency that you can see now after exposure. You just need to look at the numbers of opportunistic infections, and if you look at the reactivation of Epstein-Barr and other shingles, herpes viruses, and other retroviruses, it's all sky high. Today I got a message from our lab where we are producing these lateral flow quick tests for spike, and they were examining all the blood samples that came from Magdeburg, from Professor Koenig's laboratory, and they just for fun, they checked them for EBV. 97% of all the blood samples where we could find spike were also positive, highly positive on Epstein-Barr. So there's a reason why this is happening, and one of the reasons is the massacre in the immune cells and the disabling of the immune cells through spike and virus.
And I wanted you to just take a moment and just show us that test again, because these tests are very, very important, because a lot of people need to know whether or not they are actually positive for spike protein. Just show us that and tell us how that works.
I don't know. That's not good with that camera here. I have a slide with it, and you can see it. It's a blood prick test, so you can. They can't be used by end consumers yet. They have to go into the hands of a doctor or practitioner that is allowed to take blood, and then you pour two drops of blood on the test, and then you have a buffer liquid, and then it will tell you, it's a binary test. It tells you if you are positive or not for spike. We get to that later. And so
Okay.
there'll be, I have the first samples now for validation with Professor Koenig, and then after we validate those, I can push the button, and then we have a lot of those at disposition, and it's an excellent tool for a bigger survey to see, to get data how many people are having spike to start with. Nobody knows.
Yes, excellent, excellent. And so similarly, in terms of this here now, the SARS-CoV, the spike protein again is causing these lymphocytes to clump together and form Syncytia, like that big egg that you showed.
Yes, sir. So again, leveraging the damage everywhere you look, especially also in these cells. On the next slide, you see the same, and there it's with monocytes and neutrophils, again forming multinuclear cells. And that is all, these are all zombie conglomerates of dysfunctional, highly inflammatory cells. You don't want to have much of those. And then comes already the first interesting slide here. These are these PBMCs. And you see there's lymphocytes, monocytes, natural killer cells, macrophages, basophil, lymphocyte T cell, B cell, etc. They belong to these cells. And they, we don't have yet, that is another research project that needs to be done to distinguish exactly and quantify which of those cells contain how much spike. We can only tell in total any of those or all of them together are having spike. And here is an example. Below that you see spike protein in immune cells, 1,000 picogram in one. That was the first diagnostic. Was a patient, he was completely asymptomatic, a stratospheric level of spike protein in the PBMCs. And then he was very shocked when he saw that, and he went on a dietary supplement regime, and after 6 weeks, we could see his doctors could see a 99% reduction of the spike in these immune cells. So in this case, it was working well. What you can see, of course, if this is happening with a spike in the PBMCs, is a shift in the Th1, Th2 balance of the immune system, for sure.
And this is shifting from antibody production to cellular immunity. Yeah. So these are important. One of the things that people are not aware of is just how interconnected the whole presents. And it leads us into this interesting picture again. What does this mean?
I'm not so familiar with English poetry, but don't you say that one rotten apple in the basket can...
yes, one rotten apple spoils the lot.
Yeah, okay. And so that and the fried eggs we have already explained. So I wanted to, it's just like to make things more visible for the listener.
Yeah. So these represent the transfected spike filled PBMCs that are then passing it on to other immune cells and making them sick as well.
It's for all cells. Any cell that has spike protein and or virus and or lipid nanoparticle in it will have to be treated like an apple with a worm inside. You don't want to eat those anymore. They are foobar. So that is what this is about. I'll explain that in more detail.
Yes. And so this leads us into the kind of damage that happens in that some cells can be reawakened and others can't.
Exactly. Yeah, so I put up now the hypothesis that in the case, the route that we are, or the doctors that work with our supplements are taking, is to use this Achilles heel of the SARS-CoV-2 causing senescence. And you have to distinguish between reversible senescence and irreversible senescence. And later you'll see why that is of extreme importance. If we go to the next slide, you see the mechanism, the transfected cells with the lipid nanoparticle start to produce spike. And in our opinion, we could not measure that yet. These cells turn senescent on the spot. And that means that they are now irreversibly senescent because they contain spike and or other viral debris and or RNA and lipid nanoparticles. And these cells have to be taken out like a cancer cell. You can't save them. And the longer they stick around, the more problems they will cause, because they are highly inflammatory and they are secreting out a lot of senescence causing cytokines. So they transmit the senescence to bystander cells, which we see as in the next picture. So all the cells that contain spike, virus, and or the manufacturing instruction are irreversibly foobar senescent. Forget it, you have to take them out, especially the Syncytia, the fried eggs. But the cells that were switched into senescence as a bystander effect, they can be saved. That's all the ones on the right. They have been put into that same senescent state, but it's reversible, hopefully. We think so. And then on the next slide, you see then a very simpler categorization. Yeah. So any cell that doesn't contain any viral fragments, spike, or other debris can potentially be reversed from that senescent state. And the ones that do contain spike and or other viral debris have to be taken out. And that can be done selectively. But you need to stick to a certain following order in order to keep the collateral damage to a minimum. Yeah. And so I have been very happy to obtain a patent application that was not rejected and accepted over 1.5 years ago on how to go about it from the diagnostic side and how to get rid of these cells. Yeah, so I'm very happy about that, because it encompasses the process of getting rid of the spike as well as the testing. Now we get to the testing. So you want to know how much spike you have. On the next slide here, you see this was a small survey we did, I think 20-something patients. And we took blood samples before we did anything and had it analyzed. And then we took a provocation capsules, which will break up some of these senescent cells. And look what happened. 2 hours later, all of a sudden, the negative, the blue bar, went from 69 to 15. That means if we wouldn't have done that, you would have 50% or more false negative. People would say, oh, I'm fine, I have no spike. Yeah. But it is.
Yeah, that is a very, very important point. So let me just see if I can get this right. What happens is that a number of people have spike protein in immune cells, which are long-lived. And even though they circulate, they are largely hidden from the testing mechanism. So if you then just simply test without using provocation to make these immune cells release them, you will have a significant proportion of false negative results where you think that you are negative, but in reality, you still have lots of spike protein in the immune system that is present and probably disabling it. And does anybody else do any of these provocation tests?
Yeah, the therapists and doctors that we work with, but it's not only the PBMCs or the immune cells; it's in all cells. As we have seen before, spike is persisting everywhere, especially in the endothelium, epithelium, and other body regions, and other organs, and kidneys, and liver, thyme, just name it. So this little provocation will not only knock them out of the PBMCs, it will mobilize them also from the endothelium and from other cell types and other organ regions. So it's a good way to show, first of all, that the proof of concept for the detox, or the ability for the body to get rid of the spike, seems to work. And second, that you now have a good diagnostic tool and you can see what's happening. And the next slide is only one example that shows you now what we see most. So this is the same patient. You see ID kit before, after. That means the upper row was before provocation, very little spike, actually below the detection threshold, and the spike vaccine was not, the vaccine spike was not detected, ND, not detected. 2 hours after provocation, you see a 5-fold increase in spike from the virus. And actually, all of a sudden, we see the spike from the vaccine showing up. How did that happen? Yeah.
Yeah, so it in effect, this is hidden away inside the cells, and therefore needs propagation to get it out. But when you think about it, one of the questions that could come to you is, okay, you are getting more positive results, but is it still, even with the increased positives, still not fully identifying all the people who have persistent spike?
Of course, you're never 100%, but we do it as good as we can. And we are on completely new territory here globally. I don't know how many other people are working on this, but I don't care. Look, we at the moment have at least a tool to show there is spike here, and we are observing now and doing some surveys and trying to get rid of that. Because look, like I said before, you cannot start any sensible, smart, or successful therapy on top of an existing toxin that is persisting in the body. And so you want to get rid of that. And after, nobody has really achieved that yet. We don't know. You look at how many papers have we now gone through over the years now, thousands, showing which biomarkers are elevated in post-vac or long COVID. And we have hundreds and hundreds of diagnostics and proteomics and epigenetics and so on. But to be honest, how many, if we say that maybe 50% or more of the people that got examined had persisting spike and virus, then all these markers are taken in the presence of spike. So what happens if you get rid of the spike? Then these markers should change, but they won't. It doesn't automatically imply now a complete cure. It will just take away some of the problems. And now is the question, which ones? So,
Yes,
yeah, so when we are now the next step is now to examine before and after all these markers again, which is very expensive, and look at what has been changed to the better by taking out the spike. Nobody has done that before because nobody was able to get the spike out before. And then you have a homework list, what needs to be done now after the detox.
yes. And so what is this here showing?
That is a statistic that we saw before. You see the left bar is the blood samples that were examined without provocation, without provocation. And then the right bar next to that, all these stars, you see everywhere there's a star is a positive now. So, I mean, it's significant. Okay, it's only 13 cases, so we don't know how it looks like when you go for a couple of hundred statistically, but I think that the trend tendency is clear. Yeah? And also we had a colleague from the US and we were at the lab of Brigitte, and then we took a blood sample from him in January this year. And you see he had spike in exosomes, 235 picogram, which is already high. And he had some spike in the immune cells. Yeah, you see that on the upper one. Then he took some of these nice capsules. And 2 hours later, his spike in exosomes went up to 1100. Yeah? So that's like a piñata. You know that game for the children where they hang up this ball with all the chocolate inside and the sweets, and you hit it with a baseball, and then it breaks open. That's what happens when you hit these cells with these spike in the exosomes, because it's getting already pre-produced. And when you hit
And
that,
so what about the spike protein in these PBMCs?
they have
Were now negative.
Yeah, and that was really setting us off. And now we had already several outcomes exactly the same. And it seems that we are knocking them out within these 2 hours.
Mm-hmm. Yes. And what is this here showing?
Yeah, so this is, give me 1 second. We have to.
This is really valuable information, Joachim. The extent of the research you have done and the benefits this is showing with managing the way how the immune system gets rid of spike protein, and then the critical thing as to how this can be bound and removed to give the body a chance to recover. As we come to a close here for this section, we'll have to do another section in the future. But when we come to the close here, Joachim, people are desperate to know if this is something they are affected with. What do they do? Do you actually have tests available at the moment?
We have just finished the preliminary production of samples that now have been validated, but we are going to, we are German, so we're going to validate them again at the laboratory with Professor Brigitte Koenig at MMD in Magdeburg. So I hope that by Wednesday, Thursday next week, I can drive there, and then we have blood samples that were already examined with her methodology, and we know exactly which one of these stored blood samples contains how much spike protein in which kind of cells, and the quantity, of course. And then we will see if the test is really accurate. And when we see that, then I can push on the button of production, and then we will provide, in each pack will be 2 of those, so that you can do one test without provocation, just see. And if you're positive, you don't need to do a provocation. But when you're negative, you can, just to be sure, you can do a provocation and wait for 2 hours and then redo the test and see if you are positive then. It's not a quantitative test, it's just a binary positive or negative test. We have a certain threshold level of detection, and if that is surpassed, you have a considerable amount of spike that you should maybe address if possible. Yeah.
So this is so for the average person out there who is concerned, you know, they want to understand if they have spike protein in their blood system. You can imagine there are a lot of hypochondriacs out there who are then going to panic if they have a positive blood test.
Yeah.
But this is not supposed to make people afraid. This is supposed to give them the awareness and the knowledge that there is something that can be done about this.
Yeah, there is several reasons why I'm going this route. It was a hard way to get here. And my colleagues, and I thank them all for their dedication. First of all, nobody knows the real percentage of the population walking around with spike protein persisting in their organism. As we have seen, we in the next recording, we're going to show the effects of what can be done, what is already published now, as a result of getting rid of this spike protein, at least in some patients. In parallel, you also see the significant reduction of the microclots at the same time with the same dietary supplement regime. We have at the moment several requests from the US. They want to go into surveys and compare different kind of dietary supplement and pharmaceutical interventions in regards to spike persistence, and I'm insisting that we include also the microclots and also some other ongoing what we saw with the bacteria, because I think these are the 3 underlying big problems that need to be resolved first. So a survey tool like a quick test can be very helpful in pre-selecting patients that participate in the surveys, and also when you send in blood, especially in Germany now for this examines is expensive. So people are shy to take a blood sample and spend $300 for a test. We do have another possibility in the US already now with a mass spec from Italy. But there is still some ambiguity to the results, and I'm not so happy with it yet. We need to cross-validate again. So the reason why we want to put out those is, first of all, to make a survey, how many people are affected. That is of great interest, especially to the US administration. They are the only ones that seem to care. And so that is one thing. And the next one is also then doctors, if they have a patient in front of them and this patient has symptoms that are difficult to explain and that could be caused by a persistent spike, a test like that would help him to include different treatment modalities for that particular patient.
So just to make it clear, so somebody watching this goes, you know, I want to be tested. What do they do?
There is on the last slide of that presentation, you have my email from the healthcare channel, which my colleagues are screening through. It depends where you are. So this test will be certified for the European Union and for the UK to start with. But I think that we will get an exception in the US to use it for scientific purposes, for a survey, for a study, because it will take time to get it legalized in the US as a test that can be openly used. So it depends on the region where you live. So UK and the European Union will be a lot quicker, within probably a few weeks.
And so therefore, if they email at the link I have just put up here,
yes,
if they send an email here, we will be able to know their interest, and potentially if you have enough samples or tests out there for them to use, they can become part of that initial test. But this is not a diagnostic test per se. This is just purely part of that research exercise.
Scientific program first. I might distribute them widely, also here in Germany, to doctors, and we have a large number of therapists and doctors already working with us, and so we can have, because you don't want to only test people that are symptomatic and are already ill. You want to know better, like a survey over the general population. You want to have a random selection of people that do the test, because we think that it's more widespread than most people think, and it would be good to first have that data now, and then based on that data we can decide, and also authorities can decide what to do next.
Excellent. I think this is tremendous, Joachim. And as we close this out, I just wanted to take the opportunity to thank you for your diligence, your determination, your willingness to sacrifice time and finance to try and find solutions for others when so many people are looking the other way because of a scientific narrative. Time will tell, Joachim, that this was and is of huge value to society and the world. And I wanted to take the opportunity to say how appreciative I am for your work. Thank you very much. And as we said, I'm showing the link again to everyone. If you are interested in joining the journey, all you have to do to participate is to take a sample. All the work has already been done, and we hopefully will find something that could be of great value for the rest of the world. Thank you very much, Joachim, and we look forward to talking with you again about some of the solutions that you have found in this research journey.
Thank you all so much for your patience, and I hope that we go together on a journey of betterment. That is why we do all this. Thank you also for all your work, Philip.
Excellent. Thank you. And if you don't mind just waiting with me until after the outro. Have a good evening, everyone, and we will continue to keep you updated on this. Thank you.